Welcome back to cell biology class, everyone. We've been talking about the cell cycle, those stages of mitosis, prophase, prometaphase, all the way through cytokinesis. One thing we mentioned is the mitotic spindle, those microtubules and organizing centers that help our chromosomes move. They attach to chromatids, they attach to each other, they attach to the cell membrane, and how they're involved in separating those chromatids. So the sister chromatids are going to move towards opposite poles of the cell. So when the cell divides, you have two cells with the appropriate number of chromosomes. Microtubules have an inherent polarity. On here, it's drawn as a plus and a minus. And this is just a way of showing the two different sides. This is not a charge of them. It's just telling you that this end and this end are different. So there are different chemical properties of the different ends. Here at the minus end, this is where microtubule assembly is initiated. So this is the centrosome when we're talking about the spindle. And then the end where we see a lot of growth is at the plus end. During late prophase, that's when we really start developing a lot of the microtubules. That growth speeds up a lot. You are initiating a lot of new microtubules at the centrosomes. Then when the nuclear envelope is disintegrating during prometaphase, that's when the kinetochores on the chromosomes and the microtubules are able to contact each other. Then once those plus ends of the microtubules have bound to the kinetochores, that's when the microtubule becomes a kinetochore microtubule. So if we go back one slide, here you can see the plus end of this microtubule is bound to the kinetochore. So that's a kinetochore microtubule. It is only a kinetochore microtubule because it is bound to the kinetochore. The two kinetochores are usually located at opposite sides of the chromosome. So they're going to attach to microtubules from opposite spindle poles. So when we look at this, the kinetochores are here on opposite ends of the chromosome, and so they're attaching to microtubules that come from opposite ends. So the kinetochore on this end is attaching to the microtubule from here. The kinetochore on this end is attaching to a microtubule from here. So they're attaching to opposite spindle poles. Polar microtubules are making contact with polar microtubules from opposite spindle poles at the same time. So back to the picture here. A polar microtubule from this end and a polar microtubule from this end are going to come and make contact with each other through these binding proteins. So those polar microtubules, when they start overlapping, those cross-linking proteins will bind the polar microtubules to each other. They're actually connected through those cross-linking proteins. We saw these microtubules emanating from the centrosomes, but cells without centrosomes are still able to form spindles. I think of things like plant cells that don't have centrosomes. They're still able to form spindles. They have to use a different mechanism that is going to be promoted by the chromosomes. This is going to involve the protein RAN, which is a GTP binding protein. RAN is going to bind to GTP because of a protein associated with mitotic chromosomes. And then it'll bind to important, and then that will release the proteins that promote microtubule assembly. When our chromosomes are moving to the central region of the spindle, what we see lining up at the metaphase plate, those movements are called congression. And congression is going to occur through three different mechanisms. One is that the kinetochore microtubules, those microtubules attached to the chromosome, are going to pull the chromosomes towards their poles. That pulling force is because of the molecule cytoplasmic dynein, so pulling them towards the poles. Kinetochore microtubules are also pushing chromosomes away, so pushing them towards the center. That is because of the molecule kinesin synp-E. So kinetochore microtubules are pulling towards the poles, kinetochore microtubules are pushing towards the metaphase plate. And then polar microtubules are also involved in congression. Polar microtubules are able to sort of push chromosomes, and that is using three different molecules, chromokinesins, kinesin 4, and kinesin 10. If you're able to just label these as kinesins involved in these polar microtubules pushing the chromosomes, that's sufficient for this point. Once chromosomes reach that metaphase plate, they're going to stay there because those pulling and pushing forces are precisely balanced. But then once anaphase hit, the chromatids are not going to stay there. Those chromatids are going to separate and move towards opposite poles. That separation is going to involve the molecule topoisomerase 2. You may remember that from Biology 1 classes when we were discussing how DNA is replicated. Topoisomerase is an enzyme that's able to break and kind of untwist and help chromosomes keep their correct shape. So that is involved in those anaphase movement of chromatids. Also, changes in adhesive proteins are involved here. Those adhesive proteins are going to hold the sister chromatids together through prophase, prometaphase, and metaphase. Those adhesive proteins release those sister chromatids during anaphase so that those pushing and pulling forces are now just going to pull those chromosomes towards opposite poles. The movement of chromosomes does take energy. Moving them to separate poles uses the different motor proteins, and they're going to be actively involved in mitosis. They take their energy from ATP, and the motor proteins will change shape and pushing and pulling forces that will move those chromosomes, those attached chromosomes. Motor proteins have 3 distinct roles that they play in moving anaphase chromosomes. The first one is moving with the molecule catastrophin. So chromosomes are moved, kinetochores first, towards those spindle poles during anaphase A. We talked about anaphase dividing into A and B. During A is when chromosomes are moved, kinetochores first. That is because of this molecule catastrophin. Catastrophin is a molecule, it's a type of kinesin that is associated with the kinetochore microtubules. There's one catastrophin at the plus end of the microtubule. It's embedded right in that kinetochore, and it's going to chew up the microtubules from there, moving the chromosome kinetochore first towards the pole. There's another catastrophin motor located in the minus end of the microtubule. It's embedded in a spindle pole, and it is depolymerizing the microtubules there. So between these two types of breaking down the microtubules, your microtubules getting shorter and shorter, which is going to pull your anaphase chromosome toward the poles. Second is motor proteins playing a role in the movement of these spindle poles. Your spindle poles are moving away from each other during anaphase B, and that is through the molecule bipolar kinesin motors. There's bipolar kinesin motors or proteins that bind to the polar microtubules and are going to pull them away from each other. So this polar microtubule starts sliding this way, this polar microtubule starts sliding that way. And as these poles are moving away from each other, that's also going to result in movement of this whole structure towards its 2 separate poles. Then cytoplasmic dynein is going to be associated with the astral microtubules. Those are these little ones that kind of form the star shape. Those are our astral microtubules. So it's attached to these astral microtubules and attached to the cell cortex. Remember, that's this lining of your cell membrane. So the cell cortex is that layer of actin microfilaments along the inner surface of your plasma membrane. So as the dynein is moving those astral microtubules, it is pulling the spindle toward the cortex, helping those 2 poles move away from each other during anaphase B. Okay, so that's an interesting discussion we had about how our microtubules are involved in our ability to go through the cell cycle. We're going to come back and talk a little bit more about how our cells are dividing following this. I'll see you back for our next video. Bye.